Hi, everybody. Dr. Carey here again. We have a genetics mini lecture to address this session. And when we talk about genetics, we're going to just give a really brief overview of the very basics of the genetic content, because genetics is actually extremely complicated and you need to take a lot of extra classes in order to master components of genetics. So we're going to give you a little bit of an overview so that if you hear some of these words, your patient has some of these diseases and you're looking it up, the basics are going to make sense, okay? But just remember that you'll need to add to what you learn in this content. So to introduce our topic, we're going to talk about genetic code. So when we are talking about genetic code, we're talking about amino acids and nucleotides. Nucleotides are found in the DNA and they're organized into codons. The collection of codons is called genetic code. And when we take a look at our genetic code, you can see the double helix here is the DNA like you've seen on TV probably. If we just zoom in on just this little bit here, just this little bit in that circle, we'll see that there are letters that we've designated to different nucleotides. And these will help us interpret what the code means. So we can see that these three bases are one code for one amino acid, but they're in different orders, so they're different amino acids, right? Each codon should have 3 nucleotides, and that tells us which amino acid it is. There can be several different combinations, so if you have 1, 2, or 3 nucleotides, you could have many different combinations. The 3 nucleotides or 64 combinations is most suited for the 20 amino acids. But what we see is that we have mutations that occur, and that's what causes disease processes in the body to happen. So a mutation is when an unpredictable change in the structure of the DNA has occurred. And it usually occurs, in one little tiny segment or 2 or 3 tiny segments. Most of the mutations that occur happen in the somatic body cells and aren't passed on for one generation to the next. Only the mutations that happen in the gametes can be inherited. So this is where we get our inheritability, when mutations actually occur in the gametes. So for an example, if we look at a P53 gamete, we can see that on the left we're going to see a normal P53, and on the right it's mutated or shown with this broken crunch bite taken out of it. If it's normal, we have normal cell cycle that happens: cell cycle arrest, DNA repair, restart, apoptosis, and it moves and moves and moves in a continuous stream. When we have this broken P53, our cell cycle continues. There's no DNA repair or cell cycle restart, or even the pre-programmed cell death. It's not occurring. We're just having continuous cell cycle production. And because of that, our cells can become cancerous. The mutated P53 doesn't arrest the cell cycle. It doesn't stop it. It continues to divide, and that is what can cause the cancer. So that continual division, then our cells become accumulated, and whatever cell that is becomes enlarged inside a liver or kidney or the brain, wherever that happens to be. So when we're talking about things like whether cancer is hereditary or whether it's sporadic, so you might have heard, it's been a few years, but we've had a couple of different movie stars who had a gene for breast cancer, and that's the BR gene. that we'll talk about later on. But the breast cancer gene, if you have the money to pay for genetic testing to see if you have that gene, you can actually have your breasts and lymph nodes removed at an earlier age than when breast cancer typically begins for those with the genetic breast cancer. So if it's hereditary, you're going to see that here we're going to see that this person had it, and we also see that this person and this person had breast cancer. This person might not have. But you'll notice this person didn't because she's actually married in. So this is a male. These two people had a boy child. And then these two people had 3 girls and 1 boy, and 2 of the 3 girls had breast cancer. Breast and ovarian cancer can occur in the same individual as well. We could also see male breast cancer in a hereditary form, and usually they have a background of Ashkenazi ancestry. In the next case where it's sporadic, you're going to see here, these are family members, but in this family member and this family member aren't really related. They're married, these two are married, but they're not related by anything other than marriage. And so the breast cancer diagnosis is sporadic because we're not seeing it fall through the family line. Typically, breast cancer is diagnosed before 60 years of age, or after 60 years of age, I'm sorry. Nobody has ovarian cancer, and there's no clear pattern on one side or the other. Other genetic traits that you might see that come through are things like hair color and eye color. So we can find that we have an eye color gene and a hair color gene, and we can trace that through our family trees. If we use this example, and we can see that both parents in this example have heterozygoic alleles, so one of each, right? One capital B, one lowercase b. So that's heterozygoic alleles for brown and blue eye colors. So the capital B is dominant for brown, and the blue is recessive. Either of these alleles could be inherited by the offspring of each parent. The Punnett square diagram is going to show all the possible combinations of alleles that can be inherited, and can show the result. The children of these two parents have a 75% chance that the offspring will actually have brown eyes. In order for the child to have blue eyes, they actually have to have 2 of the recessive alleles. So brown eye color is dominant, and because of that, that's what causes this allele to result in 75% chance or probability that the child will have brown eyes. Now, what I think it's important for you to remember, and anytime we're talking about these chances, it means each conception, not each child. Or it's not going to be 75% of the kids. It's each conception has a 75% chance of having brown eyes. And it's slightly something different than saying 3 out of my 4 kids should have brown eyes. Just slightly. So we also need to talk about karyotypes, because these are the actual chromosomes that an individual has. We know that typically we have 22 chromosomes and then a pair of sex chromosomes. And normally those all come in pairs, and we will have 2 chromosomes with each pair. Not a big deal, right? In someone with Down syndrome or an altered karyotype, we're going to find that we either have 1 extra or 1 missing allele on one of the chromosomes. So in Down syndrome, they have an extra chromosome at an extra allele at chromosome 21. So we also can see that each of the chromosomes are comprised of two chromatids, and these are parts of the DNA protein strands. And when mitosis is going to begin, the chromatin packs even more tightly, closer together, so that it's more visible. The difference between the male and female, both males and females, have 22 chromosomes. But when we look at the sex chromosomes, a male has one Y, or one X and one Y, whereas a female has two X's. And that's what makes the change for a male and a female. The difference between homozygous and heterozygous, the homozygous trait is made up of two of the same alleles. So they're either both capital R's or dominant, or lowercase r's to be recessive. A heterozygous trait is made of two different alleles. So that would be a capital R and a lowercase r. That's heterozygous. Homozygous can be either two capital R's or two lowercase r's. And these are the alleles, one form of a gene. So when we see another way to show the heterozygous or homozygous is to show the alleles on pairs of chromosomes, and this is just one way we can do that. These are heterozygous because there's two, one dominant and one recessive. These are both homozygous. This one here has two dominant, and this one has two recessive. So as we look at the chromosomes, oftentimes we'll mark the alleles on the chromosomes to look like bands, and so you can see them in this way as well. The Punnett square is actually this diagram, and it of course was discovered by Dr. Punnett years ago. It's an abstract way to show how two genetic carriers combinations might impact their children, and so it's important to kind of pay attention to how that might happen. This is the same example really that we had earlier with different letters. So we have a dad with a dominant and a recessive, and a mom with a dominant and recessive. And so the chance of having a child with two copies of the recessive gene, so blue eyes perhaps in this case, is 25%. Remember, 25% for each conception. And then you've got the chance of having the dominant at 25%, and then in this case you've got this chance of having one of each at 50%. So when we talk about the characteristics that are denoted by these black circles in this diagram, remember, if it's a black circle, that's the person that shows the disease process. The white circles do not. And so in this case, we're going to see that it looks like this is a dominant. It's not sex linked because we have both male and female that are impacted. And it appears to be both dominant with both mom and dad. So if we use the same diagram to determine whether it is homozygous dominant or heterozygous dominant. So are the parents both homozygous dominant or homozygous recessive, or do we have one of each? I'm sorry, I reworded that wrong the first time. Both are homozygous recessive in this case. Then with blood type. Now blood type quite frequently you've seen the chart that basically says if parent A has A blood and parent B has B blood, then the child could have any type, right? But what you have to also remember is there's a genetic component that comes to play in here. So you also have to pull the gene characteristics in. If one parent has A blood and the other parent has B blood, then what we're going to do is actually cross off all the A's and B's, and that leaves us with type O. So we have A blood for father, B blood for dad. They could have any of these. But because father has A and mom has B blood, we can cross these off, and our patient, our child, will have O blood. That's because of the characteristics that are being exhibited in the genogram. A good question on this is actually what happens if the opposite is true, and our mom and dad are homozygous, then you're going to find that we don't quite have enough information. We need to be able to see a little bit more information. The genotype versus a phenotype. The genotype is a combination of alleles an organism inherits from its parents. A phenotype is the organism's actual appearance. So I might inherit my genotype from my parents, but I may not necessarily show that characteristic. So for the widow's peak, I might or I might not have that characteristic, even if I've inherited it. So how that trait actually exhibits itself is important. So here we are, we're going to talk about a sex-linked. So we have an X-linked dominant allele, and that's in dad. And mom has a recessive allele. So she doesn't have the disease, but dad does. So dad's affected, he has the disease. Mom is unaffected, she does not, but she's a carrier. So we call mom a carrier because she has the gene, but she does not have the disease. But because she has the gene, she can pass this on to her kids. So the father, because this is X-linked, can pass this on to his daughters. Okay, it's X-linked, right? So he can pass it to his daughters. The mother can pass it to all of them because they all have at least one X. The girls only have X's, so the dad can only pass it to the girls. So here's another example, hemophilia. Dad does not have hemophilia, but mom is a carrier of the gene. So we have this extra X-linked, and when you look at the Punnett square, you're going to see where that extra allele is coming across and where those percentages lie. So for this couple, 50% of the sons will have a chance of having hemophilia, and 50% of the daughters will be a carrier. So each conception, if it's a boy, they have a 50% chance they'll have hemophilia. If it's a girl, they'll have a 50% chance of being a carrier. When we talk about penetrance and expressivity, we are talking about the influence of a phenotype on a particular genotype because of the background of environment and genetics. So when we look at penetrance, we are wanting to see how extensive this phenotype has penetrated the genotype, And expressivity has to deal with the extent of that phenotype in a population. Sometimes we'll have multifactorial traits, and that involves multiple issues or problems. So diabetes is called a multifactorial trait, cleft lip and palate, and also clubfoot. So when we see some of these multifactorial traits, what we are looking at is a single nucleotide polymorphism, or a SNP. And just this one can be impacted. And instead of having that normal shape and function, it's slightly altered, it's slightly different, and it can't connect as it normally would. And so we'll see this change in mitochondrial DNA. It's found, it contains genetic material only from mom, where nuclear DNA is found in the cell nucleus and contains genetic material from both parents. We can have another process that's called an oncogene that can disrupt or cause a mutation. And an oncogene is basically an encoded protein with an altered structure which has changed its function. It's a new copy, it could be overexpressed, it could come from a retrovirus, and the retrovirus activates. But somehow this gene has become activated and is now an oncogene because it's going to cause a cancer to start. So we find that because of this mutation or deletion, where am I at? That a oncogene is encoded and encodes a protein. We can then see that the DNA sequence is then changed and translocated from different sites, and it alters how that gene works as it moves down the pipeline. We'll see synthesis of that protein, and fusion of the protein is no longer under control. And so that protein coding gene fuses with portions of the gene that causes this formation. We also have tumor suppressor genes that act like a brake pedal in our body. It's supposed to stop these things from happening. A common example is with growth factor, and we can stop that growth factor from making those cells grow excessively. It can happen at varying time points in that signaling processing phase. We could turn it off, or we could turn it into hyperdrive. And the hyperdrive just makes it work all the faster. The normal mechanism in our body is to shut it off and turn it down and stop those prolific cell types from expanding even further. But sometimes our bodies can't do that on its own. We can also have cancers or viruses that can cause cancers, and there's actually quite a few of these that are out today, and we have at least one vaccine. So when a virus exists that might cause cancer, it integrates into the system, and that causes some type of activation or inactivation of the oncogenes or the tumor suppressors. The genes then express themselves differently to alter that signal pathway. And then we'll have a activation of the chronic inflammatory pathway. So we have in this image a picture of a cervix, or multiple pictures of a cervix. This is the HPV virus. And there's now an HPV vaccination that's available for young people that can stop this from occurring. Herpes simplex, and I don't know what this one's called, but it has to do with overgrowth of skin cells, possibly warts that are causing those viruses. So let's take a look at this question. Which statement concerning a pair of alleles for a gene controlling a single characteristic in humans is true? A pair of alleles for a gene controlling a single characteristic in humans. Both genes come from the father, both genes come from the mother, one gene comes from the mother and one comes from the father, or it's random. It could happen anyway. So because we're talking about a pair of alleles, one allele comes from mom and one allele comes from dad. It doesn't matter what disease it is, that is how the genetics works, right? So one from each, one from mom, one from dad. The appearance of traits expressed by genes in association with environmental influence is referred to as the organism's genotype, phenotype, karyotype, or genome. So this is the phenotype. So these are the traits expressed by the gene. It's what our person looks like, why they have a cleft lip or cleft palate. This is the phenotype is what's causing that. The human papilloma, I cannot say it when I'm looking at it, but let's just call it HPV. HPV virus has been implicated in causing cervical cancer, genital warts, liver cancer, or prostate cancer. And it has been implicated in cervical cancer. Males can also get HPV that causes cancer, and it will cause testicular cancer in males. And that's the biggest difference. That's the end of the genetics mini lecture. Please let me know if you have any questions.